All right, for Shigella. So for this lecture video, we will be focusing on Shigella species and Yersinia. So let's start with your Shigella. So for your Shigella, this is still a genus with under classification of your Enterobacteriaceae. And as I have discussed a while ago, your Shigella is closely related to your genus Escherichia. Again, your Escherichia coli is the most versatile bacterium, which has the capacity to receive genetic makeups from other bacteria, and that includes Shigella. So Shigella and your bacteria shares quite similarities with each other. So what are the differences? So here are some of the differences. If you have remembered, Shigella is one of those non-motile bacteria under Enterobacteriaceae. If you have remembered, what are these three bacteria that are classified non-motile in Enterobacteriaceae? Those are your sky. S stands for Shigella, K stands for Klebsiella, and Y stands for Yersinia. Aside from that, Shigella is also capable of invading and living inside the intestinal cells, meaning Shigella, just like her Salmonella, has the capacity to hide and live inside the cells. That would actually make them intracellular microorganisms. Intra means inside. So they have the capacity to live inside the intestinal epithelial cells. And aside from its non motile, again, Shigella does not have flagella. It does not have peritrichous flagella, unlike the other species of Enterobacteriaceae. So, right in front of you are some of the important clinically significant species of Shigella. So, they are subclassified into four groups. We have the A, B, C, D. So, group A stands for Shigella dysenteriae. Group B, Shigella flexneri. Group C, that would be Shigella boydii. And Group D, that would be Shigella sonnei. Shigella sonnei, if you have remembered, has a distinct characteristic, right? So Shigella sonnei, remember that your Shigella, all of your Shigella are mostly non-lactose fermenters. There are usually non-lactose fermenters with the exception of Shigella sonnei. So your Shigella sonnei, if you have remembered, is classified under late lactose fermenter. That is together with your Salmonella arizona. So please do not forget that one. Those two important bacteria are very important with their distinctive characteristics based on its ability to ferment lactose. So again, your Shigella, going back to Shigella, your Shigella is divided into four main species. So each main species has different groups and it is associated with different diseases as well. So your group A again is your dysentery. So dysentery because it is known to cause severe dysentery. So we have here flexneri. It is very common in developing countries and also causes moderate dysentery. So we have also bodii, which is group C. It is less common, but then again, it could also cause dysentery. And Sonny eye, this is the most common cause of shigellosis, which is very common in industrialized. When you say industrialized, these are in most developing countries. If you have remembered once again, Shigella is the most common culprit for bacillary dysentery. So it is the most common culprit for bacillary dysentery. So how do we actually get Shigella? Shigella is primarily transmitted via the fecal oral route. So via fecal oral route, which can occur through the four F's. We have through F, the flies. So that is your biologic vector, flies. So flies can actually carry Shigella bacteria from feces to food. We have also fingers. So contaminated hands can transfer bacteria to food or mouth. We have food. So we could actually consume contaminated food, which can introduce bacteria into our digestive system. And last but not the least, we have also feces. So, feces and water. So, drinking water may be contaminated with feces or coming in contact with infected feces. So, these are some of the most common causes or sources of the bacteria. So, just like your Salmonella, Shigella has a human only reservoir, meaning that is usually found this bacteria is usually found solely in humans and does not have an animal reservoir, meaning the main transmission of this bacteria is from person to person. Therefore, the infectious rate is usually high compared to the other bacteria. So, Salmonella and Shigella, again, human reservoir only. So, it does not have any animal reservoir. So, right in front of you is actually the classifications and subgroups of your Shigella. So, what we have here is Group A, B, C, and D. We have dysentery, flexneri, body eye, and sunny. So we have here D, flex, boy, sun. So we have also other names. So dysentery stands for the Shiga bacillus. So it usually causes again dysentery. We have flexneri, flex, Strong's bacillus. Body eye, we have the Newcastle Manchester's bacilli and the Sonnier, Son ducal bacilli. So Sonnier again is the most clinically significant bacteria among all the subgroups presented to you, and that is group D. So that is classified under group D. So the primary virulence factors of Shigella is the Shiga toxin and its antigenic structure, which is somatic O. If you could remember, Shigella is non-motile, so therefore it has no H antigen. It is non-capsulated, therefore it doesn't have the K antigen. So for Shigella, the antigenic structure is only the somatic O, and we have also the Shiga toxin. If you have remembered an E. coli structure, we have this type of strain of your E. coli that mimics your Shiga-like toxin, right? So if you could remember that one, we have our Shiga-like toxin. It causes your hamburger's disease, so that would be your EHEC. So your STEC, so Shiga-like toxin producing E. coli. So if you have remembered that one. So in here, the Shiga toxin is the potent exotoxin. So this is the main virulence factor of your Shigella, primarily produced by Shigella dysentery. So this is primarily produced by Shigella dysentery, and to a lesser extent, it is also produced by other Shigella species. So your Shiga toxins plays a critical role in the pathogenicity of your Shigella, even though we have only two virulence factors. That includes your antigenic structure and your toxin of your Shigella. Shigella causes the most one of the most severe type of diarrhea, and that is dysentery. So what is the pathogenicity of our Shiga toxin? So your Shiga toxin inhibits protein synthesis in host cells. So if you have remembered in your biochemistry, protein synthesis is very important to cells. So if there's no protein synthesis, it would actually lead to cell death. So, if there's cell death, it could actually help in contributing the symptoms to the dysentery. So, aside from dysentery, patients suffering from shigellosis may experience abdominal pain or abdominal cramps. So, other than this, the ability of the toxin is also to disrupt cellular function. That is why this is the most important factor in the severity of Shigella infections. So that is the Shiga toxin. So for antigenic structure, if you have remembered, your somatic O antigen, which is usually found in your LPS components of the cell wall, these are usually used to classify Shigella into different serological groups. So just like your Salmonella and other bacteria, your O antigens are integral part or the key factor in your bacterial cell wall that is used to identify specific serotype of Shigella. That is your somatic O. Take note, Shigella is non-motile and non-capsulated. Therefore, in your antigenic structure, we only have somatic O. So what we have here is a bacillary dysentery on your picture that would actually tell you an example of green pea soup. So that is not the real stool for patients suffering from bacillary dysentery. So that is a real green pea soup stool that you eat. But then again, if a patient suffered from bacillary dysentery, the stool of the patient may look like this. So, a green pea soup stool. Again, green pea soup stool is associated both Shigella and Salmonella. So, bacillary dysentery is also known as shigellosis. Again, shigellosis. So, bacillary dysentery is also known as shigellosis. Therefore, the causative agent of your shigellosis is your Shigella dysentery type 1. Shigella dysentery type 1. So this is the specific serotype of Shigella that is known to cause severe forms of dysentery. So your bacillary dysentery here is characterized by the following. We have here acute inflammatory colitis. What is colitis? That is the infection causes inflammation of the colon, colitis. So, if there's inflammation of the colon, the patient may experience painful and frequent bowel movements, sigig kalibang. So, the patient may experience the urge to poo after pooping. So, what do we call that? So, what do we call that? So, that is actually tenesmus. So, we have also bloody diarrhea. So for bloody diarrhea, the patients experience diarrhea containing blood or mucus, and WBC. Okay, so in the bloody diarrhea, patients may experience diarrhea containing blood, mucus, and WBCs. So these combinations of symptoms may actually indicate inflammation and damage to the intestinal lining. So I think I have already discussed to you the difference between an ordinary diarrhea and dysentery. So a dysentery, so key factors of your dysentery, it's the presence of WBCs and RBCs. So how do we get Shigella? So again, Shigella dysentery type 1 to be specific is highly communicable, meaning it is highly infectious, and unlike your E. coli that the virulence factors or the infectious load or infectious dose will range from 1 million to 1 billion, for Shigella dysentery, as few as 200 bacterial cells. Shigella dysentery can cause now infection. So how do we get this one? Number one, that is with the help of the human careers. No? So we could actually get this, not help. So that is mediated by human careers. So for human careers, so these are infected individuals again that are usually asymptomatic, but then again could spread the bacteria. So we have here the fecal oral route and the person to person. Again, Shigella is strictly person to person and a human pathogen. So it has no animal reservoir. So here are some of the basic characteristics of your Shigella. So all Shigella are non-motile, meaning all Shigella species are non-motile, meaning this bacteria has no H antigen. So unlike many other enteric bacteria, Shigella lacks flagella. So for the mannitol fermentation is very important in differentiating Shigella dysentery from other species of Shigella. So all species of Shigella are mannitol fermenters except Shigella dysentery. Again, all species under Shigella are mannitol fermenters except Shigella dysentery. Shigella dysentery are non-mannitol fermenters. So they're also negative for gas, so meaning it cannot produce gas. So we cannot observe bubbles or cracks when Shigella is inoculated in your TSI or in your LIA. So it is negative for gas. except for Flexneri. So, except for Shigella flexneri, it is Urase negative. And please remember the Imbric reaction, that would be variable positive, negative, negative. Okay, so these are some of the distinctive features that we could actually help differentiate Shigella from other species of Shigella. So, especially your dysentery and flexneri. So for TSI, it is K over A. K over A with negative gas and negative hydrogen sulfide. So again, what does this mean? So it's K over A, meaning it has an alkaline slant and an acidic butt. So what is usually found in the slant? That is your lactose and sucrose. So meaning in the slant, there's no glucose fermentation. There is no lactose or sucrose fermentation. So since it has an acidic but only glucose fermentation. So there is only glucose fermentation. So it is negative gas, so no gas production and no hydrogen sulfide production as well. So this is for the culture method of Shigella. I have discussed the difference between your Salmonella and Shigella based on the inoculation of the following augurs. Number one is your Macongay. So if you have remembered, all Salmonella and Shigella species are non-lactose fermenters. So both of them shows clear colorless colonies when inoculated in your Macongay, with the exception of Salmonella arizona and Shigella dysentery. So, Shigella dysentery. So, what we have here is your Heya. So, Heya, when your Shigella is inoculated in your HEA, so your Shigella shows colorless colonies without black centers. So again, it shows colorless colonies right here with no black centers. While your Salmonella, on the other hand, it shows colonies, growth of colonies with black centers. Again, your black centers indicates that the bacteria can produce hydrogen sulfide. So here, for your Shigella cannot produce your hydrogen sulfide. Therefore, upon inoculation with your HEA, we cannot observe black centers around the Shigella. Okay, so that would be your Shigella. So last but not the least is your Yersinia. So your Yersinia is subclassified into three important species, and that would be your Pestis, your Enterocolitica, and your Pseudotuberculosis. So what I have here is the famous nursery rhyme, Ring Around the Rosie. So I have read that this poem is often associated with the Black Death. If you're familiar of the Black Death, it was one of the first devastating pandemic that swept through out the Europe in the 14th century. And the culprit of the pandemic is a bacterium. That bacterium is what we call as Yersinia pestis. So in here, Ring Around the Rosie refers to the characteristic rash of the rose-colored spots. If you know this rhyme or this nursery rhyme, it is very famous to children, kindergarten. So we have your pockets full of posies. Why it was um, referred to pockets full of posies? Because posies on that time were carried to mask the foul odors associated with the disease, and ashes will all fall down. It actually refers to the practice of burning the clothes and belongings of plague victims to prevent the spread of the bacterium, or of the infection. So at that time, the scientists or the doctors still don't know the causative agent, and they don't know the mode of transmission. So what they have been doing is burning the patients and burning all of the belongings of the patient. So it's actually the same story what we have experienced way back 2019, that we experienced a novel virus. So we were locked down. So there was a pandemic all over. But what was the good side on our situation is the technology and science is already modernized compared to the last 14th century. So that is actually the story of your Yersinia pestis. With that story, Yersinia pestis was also known as your Pastorella pestis, and it is referred as the plague bacillus. It is referred as the plague bacillus because it is the bacterium behind the bubonic plague, and it is also the bacterium behind the Black Death. So doctors at that time are wearing this infamous mask right here. So that was a pandemic that actually causes to death to 25 to 30 million people at that time. So how do we actually get Yersinia pestis? Please take note that Yersinia pestis is very unique. This is the only enterics that is transmitted to humans by a vector, and that vector is your infected flea. So that vector right here is your Xenophila kelpis. Is your Xenophila kelpis. If you have remembered, we have this one parasite that is also related to Xenophila kelpis. If you have remembered that one, so that would be your assignment of what parasite that would be. So again, your Yersinia pestis, since it's a pestis, it is transmitted through a pest or an infected flea. So again, it is the only enterics that is transmitted through a vector or through a pest. So this Yersinia pestis is now classified under Class A bioterrorism agent. So it is a bioterrorism agent. What we have here is the identification of your Yersinia pestis. So when we look at your Yersinia pestis under the microscope, we could use specific stains to reveal its distinctive features. As you could see there on your screens, the Yersinia pestis has a very unique morphology, and that unique morphology is what we call as the safety pin appearance. So the key characteristics of your Yersinia pestis appears as a short, plump, rod-shaped bacteria. So it is short, plump, rod-shaped bacteria. So it is often again described as having bipolar staining or safety pin appearance. So we would be using stains for this purpose for us to differentiate this morphology. So the stains that are usually used are your Wason stain and your methylene blue. So Wason stain on your methylene blue. So your bipolar staining right here on your screen. So the bacteria may look, it has dark ends with lighter centers. If you could see, it has dark ends with lighter center. So it actually resembles a safety pin. So this is a way the stain interacts with the bacterial cell wall and its internal structure. So for when your Yersinia pestis is stained with Wason or methylene blue, this bacteria often shows characteristic with a rod-shaped appearance with a darker ends and a middle part and with a lighter center. Again, the middle part is actually lighter. So it actually creates a visual effect similar to safety pin. So that would be your Yersinia pestis. Aside from that, when culturing your Yersinia pestis, we could use various media to observe its growth patterns. So on your blood agar plate right here, at 24 hours at this early stage, Yersinia pestis forms a pinpoint colonies. So when you say pinpoint, these colonies are very small and may be challenging to see without careful observation. So pinpoint colonies, this is very similar to your streptococcus. So pinpoint colonies in your blood agar plate, so within 24 hours. But then again, after a longer incubation period, the colonies develop this one. This is actually a distinct rough cauliflower-like appearance. So a distinct rough cauliflower-like appearance. So imagine looking a colony that resembles the surface of a cauliflower. So it is compared to the pinpoint colony right here. This one, this is a pinpoint colony. Compared to this pinpoint colony, the cauliflower is a bit lumpy. It is also a bit irregular with rough structures. With rough structures compared to your pinpoint colonies. So this change in appearance helps differentiate Yersinia pestis from bacteria. Again, for 24 hours, it looks like pinpoint colonies. Your Yersinia pestis forms pinpoint colonies. It is very small compared 48 hours of incubation. It resembles the surface of a cauliflower, which is lumpy and irregular. Okay, so how about in your broth culture? In your broth culture, Yersinia produces a stalactite pattern. Stalactite. What does this mean? So this means that the growth in the broth forms structures hanging down like stalactites in a cave, if you're familiar with stalactites. So these are your stalactites. So this is your broth culture. Again, when Yersinia is grown in a broth culture, this means that the Yersinia produces structures hanging down like stalactites in cave. That is why Yersinia pestis is known to produce a stalactite pattern. So this unique growth pattern actually helps us in identifying the bacterium. So aside from this biochemical test, we need also to remember its virulence factors. So your Yersinia pestis has a series of virulence factors, and that includes your endotoxins, coagulase, and fibrinolysin. So if you have remembered your endotoxins are very common to gram-negative bacteria. So endotoxins can trigger severe inflammatory response if you have remembered. So this would trigger severe inflammatory response that could contribute to symptoms like systemic shock or fever. So any systemic infections or systemic symptoms, the culprit of that are usually the endotoxins. How about the coagulase? So the coagulase, the bacterium produces this type of enzyme, which again helps evade the immune system. If you have remembered your coagulase in your Staphylococcus aureus lecture, coagulase causes blood clotting. So if there's blood clotting, this could actually help protect the bacteria from phagocytosis by the immune cells. So, if the bacteria evades phagocytosis, it allows the persistence of the infection and could also cause the disease. If there's a coagulase, your Yersinia pestis also has the fibrinolysin. Fibrinolysin is the complete opposite of your coagulase. If your coagulase causes blood clotting, your fibrinolysin is another enzyme that usually breaks down the fibrin. So it breaks down the fibrin, which is responsible for blood clotting. So therefore, Yersinia pestis also has the ability to disrupt blood clots. So if it disrupts blood clots, it can actually spread more easily through the bloodstream and tissues. So this is how your Yersinia pestis do its job in um, spreading through the bloodstream, which may cause further to sepsis and septic shock. So here are some the biochemical tests. So for the biochemical test, we have the Imvik reaction. For the Imvik reaction, we have negative, positive, negative, negative. So therefore, your Yersinia pestis is indole negative, methyl red positive, Voges Proskauer negative, and citrate negative. So for your TSI, that would be K over A. So that would be alkaline over acid. This means the bacterium ferments glucose only. So it ferments glucose only, resulting in an alkaline reaction in the slant and an acid reaction in the bot. So we have also the gas production. For Yersinia pestis, it indicates no gas production. It's gas negative, and as well hydrogen sulfide negative, H2S negative, meaning no hydrogen sulfide production is happening. So that would be your Yersinia pestis. Again, please remember your Imvik reaction and your TSI reaction. What we have here are the three forms of plague. So each form has actually distinct characteristics and mode of transmission. So number one, we have here the bubonic plague. So in the bubonic plague is the most common form of the disease. So it is what often we often think of when we hear plague. So it is usually caused by high fever and swollen lymph nodes. These swollen lymph nodes are known as buboes. So these buboes are usually appear in the groin, right here, groin, armpits, and neck. So just imagine swollen lumps in these areas. So if these areas are swollen or inflamed, usually this is what we call as buboes. That is why it is known to be the bubonic plague. So, what is the mode of transmission of your bubonic plague? So, the primary mode of inflammation or transmission of your bubonic plague is the bite of an infected flea. If you have remembered, the bite of an infected flea, the flea is your Xenopsylla Gyopsis. So again, that is your infected flea. So your fleas have the capacity to fed on infected rodents, and these rodents could actually transmit the bacteria to humans. So the fleas that are usually found on rodents can transmit the bacteria to humans. So these are the common cause or the mode of transmission of your bubonic plague. Another type of plague is your pulmonary plague. So your pulmonary plague is when the plague bacteria spreads to the lungs. So this is often a complication of your bubonic plague. So this, since it involves the lungs, it involves severe respiratory symptoms, just like respiratory distress. We have cough, chest pain, difficulty in breathing. So these are some of the signs and symptoms that are commonly manifested in a pulmonary plague. So how do we get the pulmonary plague? If your bubonic plague is transmitted via a bite of an infected flea, your pulmonary plague is acquired through person to person. So, meaning it is spread from person to person through respiratory droplets. So, when an infected individual coughs or sneezes, so this means we have a close contact with someone who has a pulmonary plague, we can actually transmit the disease, making it highly contagious compared to bubonic plague. So pulmonary plague is highly contagious compared to bubonic plague. How about the septicemic plague? Among these three forms of plague, septicemic plague is the most severe. It is the most severe and it can have its own complication. So this could be a complication either of the bubonic and the pulmonary plague. So let's review its features. How do we get septicemia? So this form, either the bubonic or the pulmonary plague, this form actually affects the bloodstream. So when this either of these types of plague enters the bloodstream, it could actually cause symptoms. So it would elicit the immune response. It could cause fever, chills, tachycardia, or rapid heartbeat, shock. So a septic shock. So it could actually cause severe complications as worse as organ failure, skin necrosis, just like this one, septicemia. Okay, if you have remembered your Staphylococcus aureus lecture, TSST1, so your TSST1 also has the same complications. So we have your skin necrosis of your septicemic patient. So that is one of the clinical manifestation of your septicemic plague. All right, so what we have here are some general characteristics of your senia and how it causes diseases and how we also identify them in the laboratory. So your Yersinia enterocolitica, we're done of your pestis. Your Yersinia enterocolitica is also the most commonly isolated species of Yersinia genus, okay, of your Yersinia genus. So it is very important for us to recognize that it is a primary pathogen in cases of enterocolitis. So it is a primary pathogen in cases of enterocolitis. So meaning this same bacterium is the most common cause of enterocolitis, which is a type of gastroenteritis. Your enterocolitica is often waterborne. So since it could cause gastroenteritis, it has signs and symptoms that like abdominal pain, abdominal cramps, diarrhea, and fever. So your enterocolitica is very unique. It has one distinctive feature. It is temperature sensitive. So it is temperature sensitive. It is very motile. Lihukan kay siya if it's cold. So if it's cold, it is very active. But it is not motile when init. If init gani, alimuot, magpuyo ra sad siya. But it is not motile at the human body at 35 degrees Celsius. So remember, your enterocolitica is temperature sensitive. It is motile. It is most active at 22 degrees Celsius. It is non-motile sa mga init na situation or temperature of 35 degrees Celsius or human body temperature. So it's non-motile when we actually harbor this kind of bacteria. So therefore, with that information or fact, we could actually isolate Yersinia enterocolitica using a cold enrichment technique. So again, your Yersinia enterocolitica, which loves the cold temperature, we could use this distinct feature in isolating the bacteria. So we could use cold enrichment technique. So how do we do this? This actually involves incubating the sample at a lower temperature to encourage the growth of Yersinia enterocolitica while suppressing the growth of other bacteria. So if you have remembered, most of the other bacteria cannot grow in cold environment, and only Yersinia enterocolitica has the capacity and has the ability to be motile and to grow beautifully in a cold enrichment technique in a low temperature. So this is actually a method that that we used in isolating and producing pure isolates or colonies from Yersinia enterocolitica with the use of cold enrichment technique. So aside from this specialized technique, we could still use the routine culture media like your BAP and your MacConkey. So on these media, some of the colonies can be further analyzed. So if you have remembered, is Yersinia enterocolitica a, a non-fermenter or a lactose fermenter? So if you have remembered, Yersinia enterocolitica is a late lactose fermenter. So it's a late lactose fermenter, Yersinia enterocolitica. So when this is inoculated in your MacConkey, it can still produce your pink colonies. So your pink colonies. Aside from that, we could use some biochemical characteristics based on its ability to ferment sucrose and mannitol. So take note that your Yersinia can ferment sucrose and mannitol. So meaning they can utilize this sugar as a source of their energy. So mannitol and sucrose. Next. So what we have here are some variety of gastrointestinal issues. So again, your Yersinia enterocolitica is the main cause of your enterocolitis. Again, this is a type of waterborne gastroenteritis. So how do we get Yersinia enterocolitica? Mainly, we get this from the consumption of contaminated food. So the primary mode of transmission for this bacteria is through the consumption of contaminated food, specifically incompletely cooked food, particularly meat products as well as dairy products. So if you're fond of eating your steak um, raw or rare, no, so you could actually get this one. So this highlights the importance of good food preparation as well and cooking to prevent this infection. So for your Yersinia enterocolitica, if Salmonella and Shigella do not have animal reservoir for your enterocolitica, we could actually found this bacteria in a variety of animals. So these animals are as well acts as reservoirs for this bacterium. So we have the swine right here. So swine, we could get this. So therefore, undercooked pork can be a source of infection. We have dogs and cats. Domestic pets can harbor the bacterium and potentially transmit it to humans. So we have also rabbits. We have also cattle as well. So how do we identify Yersinia enterocolitica? Again, just like other Yersinia, your Yersinia enterocolitica has short, plump appearance. So that would be your Coccobacilli. So it has also a distinct feature, which is the bipolar staining effect, often described as your safety pin appearance. So again, this could be observed using stains such as your Wason and your methylene blue. Aside from that, we would also be using your CIN. Your CIN or your cephsolodine ergosanovobiosin agar. So this is a specialized agar that suppresses the growth of other competing bacteria and only enhancing the growth of your Yersinia enterocolitica. So what is good in your CIN? It has a distinctive, it shows distinctive morphology of your Yersinia enterocolitica. So your Yersinia enterocolitica, when isolated or inoculated in your CIN, it shows bullseye colonies. It shows bullseye colonies right here. So on your screen, on your lower part portion of your screen, so you can see here your bullseye colonies. So these colonies usually appear with a distinct center surrounded by a clear zone. So we have here a distinct center with a clear zone. So typically this is still visible after 48 hours of incubation. So just like your other Yersinia, so your Yersinia enterocolitica, the bullseye colonies are typically observed after 48 hours of incubation. So these are the biochemical tests for your Yersinia enterocolitica. For Imvic reaction, we have variable positive, negative, negative. So for TSI, so in most cases, by the way, for Imvic reaction for your indole, for Yersinia enterocolitica, it shows a positive result for indole. But then again, we have certain conditions and situations where it shows variable results. Meaning, when you say variable, it could be positive, it could be negative. But then again, for Yersinia enterocolitica, it shows more on the positive side. So we'll just put variable there for variable positive, negative, negative. Again, for INVIC reaction, INVIC is not the only biochemical test that you need to perform in order for you to verify that what you are having is Yersinia enterocolitica. So INVIC are just one of the series of biochemical tests that you need to perform. So your INVIC reaction should be supported with other tests, just like your TSI. So for your TSI, for your Yersinia enterocolitica, that would be K over A. That means we have an alkaline slant and an acidic bot. So meaning only glucose is fermented, negative gas and negative hydrogen sulfide. So last but not the least is your Yersinia pseudotuberculosis. So Yersinia pseudotuberculosis is a bacterium that is pathogenic primarily to rodents. But then again, it can also has an effect to humans. So we'll cover its primary reservoirs, modes of transmission, and some key biochemical characteristics to understand how it causes disease and how it can be identified in the laboratory as well. So since it is very common to rodents and some guinea pigs, it has reservoir. It has an animal reservoir. So the usual animal reservoir are the guinea pigs. So it could actually cause a range of symptoms of these animals, which we will not discuss anymore since we're not vet med students. But then again, this bacterium, especially if you're fond of petting guinea pigs, so these bacterium can be sometimes be transmitted to humans. So then again, your Yersinia pseudotuberculosis, the reservoir are the guinea pigs. So, but then again, it could also be transmitted via close encounter to these reservoir. So you could actually harbor these pathogens in their bodies. So mode of transmission, very self-explanatory. Contact with the infected animals and as well ingestion of contaminated food. So another mode of transmission is the ingestion of food contaminated with this bacteria. So usually the contamination can occur if the food is improperly handled or if animals shedding the bacteria are in close proximity to the food sources. Say, for example, if you have your guinea pig pet with you, if it is closely um it is in close proximity to your food source, it could actually shed the bacterium to your food. So you could actually get that through ingestion. So your Yersinia tuberculosis is also motile at temperatures ranging from 18 to 25 degrees Celsius. So they're also motile at this temperature. So they're also temperature sensitive, just like your Enterocolitica. So your temperature, this temperature range would actually reflect the ability of the bacteria to thrive in cooler environments. So, this is very typical for many environments and animal associated bacteria. So, aside from this one, the most common test for Yersinia tuberculosis is the urease test. So, the bacterium produces urease. What is urease? If you have remembered your clinical chemistry, this is an enzyme, urease, that hydrolyze your urea. So it is urease. So it's an enzyme. It would hydrolyze your urea into two products. What is that? That would be your ammonia and carbon dioxide. So this is a very unique characteristic of Yersinia pseudotuberculosis that can be differentiated from other similar pathogens. Please remember that one. Pseudotuberculosis, Yersinia pseudotuberculosis, urease positive. And other than that, your Yersinia pseudotuberculosis also ferments this type of sugar, and that is rhamnose. Okay, so right in front of you is the difference between or differential test between your Yersinia species from your IMVIC reactions down to your temperature sensitivity tests to your urease and then your enzymes and the ability of these bacteria to ferment sugars like your lactose and sucrose. So please take note of the following. This is found on your Bailey's and Scott's. All right. So I guess by now you're already done with your opportunistic pathogens of the family enterics discussion. So we will end our lecture here. Thank you very much for listening. I hope, actually, I have here love story, but then again, so we don't have any time for that. So I will be uploading the video of Mary Malon on your Batch G drive as well. So this would be my references that would be made